Hello everyone, welcome to Ritika's online teaching academy. Today I will talk about the DNA replication in prokaryotes. Earlier in the previous lecture I have talked about the general DNA replication, what is replication, how it takes place and what are the different enzymes involved in it. So today in detail I will discuss about replication in prokaryotes. So I'll share my screen with you. Okay. So this DNA replication in prokaryotes is part 2 of molecular basis of inheritance and is lecture 4. So first we'll recall the general features of DNA replication. As we all know that DNA replication is semi-conservative in nature. What does this semi-conservative term stands for? It stands for that the one in the newly synthesized DNA molecule, one would be of the parental DNA strand, while the other strand or the complementary strand belongs to the new formed strand or new daughter strand. Now, the second feature is that it always begins at the origin of replication. Means for DNA replication, a particular site or point is required, which is ORI or origin of replication. Third point is DNA replication is bidirectional. Bidirectional meaning in earlier lecture I have talked about it, that it will move in two directions, in both the directions from the origin of replication. Okay, now the replication starts. In order to start the replication, the DNA molecule, which is a double-stranded DNA molecule, it must have to unwind, or the two strands must be separated from each other. So these two strands are antiparallel and complementary strands, and they result into the formation of two single strands. This unwinding results into the formation of two replication forms. Okay, so when suppose for an example, if you take a rope and pull apart its two strands, then what will happen? A bubble-like structure would be created, and at the two ends of the bubble, the structure which is formed is called as a replication fork. Then the replication always proceeds in 5' to 3' direction and is semi-discontinuous. Okay, means on one strand, parental strand, DNA synthesis would be continuous, while on the second strand, DNA strand formation would occur in small pieces of DNA called as Okazaki fragments. Hence, it is lagging strand and is not continuous. Hence, the whole process of replication is called as a semi-discontinuous. So DNA is synthesized by DNA polymerases. So DNA polymerases are the main enzymes which are responsible for the formation of DNA. Of the two newly synthesized strands, one is synthesized continuously in the direction of movement of replication fork and is called as the leading strand, while the other strand is synthesized discontinuously away from the movement of replication fork and is called as the lagging strand. All these points I have discussed in detail in the previous lecture. So in order to refer those lectures, you please do like, share and subscribe my channel on YouTube. Okay, now from this picture you can understand these two black colored strands belongs to the parental DNA strand. Okay, so one and I have shown here, not the other one. The newly formed DNA strand you can see here is in 5' to 3' direction. Means it always occur in 5' to 3' direction, and this red color is of belongs to the RNA primer. Okay, now the on the other strand like this. Okay, so replication fork is moving in this direction. Okay, and you can see on one strand the DNA synthesis is continuous in the direction of movement of replication fork, while on the other strand the newly synthesized DNA is moving away from the movement of replication fork. Okay, so this belongs to the lagging strand, while this one belongs to the leading strand. Okay, now you can see once this is finished, the second RNA primer now appeared, and then this black colored arrow is indicating the newly formed DNA. So these in pieces, DNA synthesis occurring in pieces called as the Okazaki fragments. So the Okazaki fragment synthesis or lagging strand synthesis proceeds away from the moment of replication fall, while the leading strand synthesis occurs towards the moment of replication fall. Now, DNA polymerases, as they are the main enzymes for the synthesis of DNA. So I'll discuss it in detail in E. coli. I am taking an example of an E. coli as a prokaryote. So in E. coli, at least 5 types of DNA polymerases are found. Means more than 5 are also there, but the main principal enzymes which have their role are 5 in number. So the first one is DNA polymerase 1. It was the first enzyme to be isolated and purified by Kornberg in 1955, and it is encoded by a poly A structural gene. So enzymes are synthesized by some genes, all enzymes. So this enzyme is synthesized by a structural gene called as the polymerase A or pol A gene. So it has two types of exonuclease activity. This DNA polymerase 1 activities are involves two exonuclease activity. The first one is 5' to 3' exonuclease activity, while the other one is 3' to 5' exonuclease activity, which is also called as the proofreading activity. Proofreading means what? From 3' and whatever the base, if it is not correct, it would degrade it or will remove it. So this activity is called as the 3' to 5' exonuclease activity. And 5' to 3' in 5' to 3' direction, it will check the occurrence of any unusual base. So DNA polymerase 2, this enzyme is encoded by the pol B structural gene, and it has only 3' to 5' exonuclease activity, also a proofreading activity only. So you can see that DNA polymerase 1 is having, these are the differences. You should know that it is having both the exonuclease activity, while DNA polymerase 2 is having only 3' to 5' exonuclease activity. So not only one enzyme is involved in exonuclease activity. If there are another enzyme is also there, if any problem is there, it would correct it. So why it is so? In order to ensure the correct replication in E. coli. Now, DNA polymerase third. This is the main principal enzyme for the DNA replication, and it is encoded by the structural gene called as the pol-C, and it has 3' to 5' exonuclease or proofreading activity also. So it is having polymerase activity as well as exonuclease activity in 3' to 5' direction. Means it is having two activities, polymerization as well as exonuclease activity in 3' to 5' direction. And its polymerization rate is very high, is about 250 to 1000 nucleotides per second. So per second it is adding these many nucleotides to a newly synthesized DNA strand. So it is more efficient enzyme for the polymerization. So DNA polymerase 4th and DNA polymerase 5th, these enzymes involved in unusual form of DNA repair mechanism. When I will talk about the repair, then I will explain about this also. Now, this is the whole structure of the DNA polymerase 3rd. Okay, so you can see that its structure is also very complex. You can see here this 6 hexameric ring of DNA B helicase. I will tell you why we are saying it as a DNA B helicase. In case of E. coli, those helicase, primase, topoisomerase, they are termed by the different names like DNA B. It functions same as that of helicase. So helicase in E. coli is DNA B. So now you can see here these two core molecules or sorry core structures are there. They are called as core polymerases. Okay, and this blue colored region is a pair of beta clamp. Okay, you can see here, and the other units you are seeing here, two tau units are present, gamma unit and delta dash. Okay, now in point wise we will understand what it is. DNA polymerase third, main enzyme of DNA replication, and polymerization rate is very high, around 250 to 1000 nucleotides per second. So we can say that its processivity is also very high. What do we mean by processivity? See, once when the DNA polymerase starts polymerizing or starts adding nucleotides to the growing nucleotide chain, or you can say a DNA chain, then it moves also along with the growing chain. So once the nucleotides are added, the replication is now that strand is finished. Then what would happen? That polymerase itself dissociate, and this rate of nucleotides adding before its dissociation is called as a processivity, and is around greater than or equal to 5 lakhs. So it completes, it can add up to 5 lakhs nucleotides before its dissociation. So it is very complex. What is very complex? DNA polymerase third is very complex and is made up of 10 subunits, 10 different types of subunits. So as we have seen here, the structure. Now I am discussing the in explanation form of each structure. Core polymerases, two core polymerases are present, and each is having three subunits called alpha, epsilon, and theta. Okay, now you can see here. In brackets, I have written alpha, epsilon, and theta. So this is a core polymerase. Okay. This core polymerase 1, core polymerase is also here 2. So both the core polymerases are having alpha, epsilon, and theta subunits, and their functions are also different. Alpha and epsilon form does the function of polymerization, while the theta alpha and epsilon have polymerization and proofreading activities. Alpha is having polymerization while the epsilon is having proofreading activity. Now theta subunit associate with alpha and epsilon to form core polymerase. So we can say that in core polymerase, three subunits are there: alpha, epsilon, and theta. Their functions are different. Alpha performs the polymerization work, while epsilon performs the proofreading activity. Now theta, after that, theta subunits are associated with epsilon, alpha and epsilon to form a core polymerase, which polymerize DNA but with low or limited processivity. Means it also has a function of polymerization but with limited processivity. So these are two core polymerases constituting alpha, epsilon, and theta subunits. Now, clamp loading complex or gamma complex, it consists of 5 subunits, which are of 4 different types: tau 2, gamma, delta, and delta dash. Tau 2, tauki, there are 2 tau present here. One is gamma and 2 delta and delta dash subunits. You can see now these two core polymerases are linked to each other with the tau subunits, with the help of tau subunits. So the two core polymerases are linked by tau subunits, and two more subunits, chi and silane, are also present, but they are not shown here. Okay. Now the third one is beta sliding clamp. Okay, one more thing. Clamp loading complex. So where this clamp loading complex is? Clamp loading complex is this one. Okay, this one is what? Clamp loading complex. So it will load a clamp, and where is that clamp? This is the clamp. So this clamp is loaded with the help of this clamp loading complex. Okay, now beta sliding clamp. Okay, 4 beta subunit associates in pairs with a core subassembly of DNA polymerase 3. It acts like a clamp and encircles the DNA. So 4 beta subunits associate in pairs with a core subassembly of DNA polymerase 3. So you can see here, see these are the beta clamps. Okay, so they are present in pairs. You can see pair, in pair they are present. Okay, so it acts like a clamp. Okay, and encircles the DNA. So this structure, this full structure belongs to DNA polymerase third. So these three structures you are seeing are having beta clams. So how they function? One beta clam would be loaded on the leading strand, or you can say the one parental strand on which the leading strand is synthesized. So it encircles that one parental DNA strand, and this encircles another DNA parental strand for synthesis of lagging strand. Okay, and this will also assist in it. Okay, now we will study it with the help of a diagram in later slides. So we can summarize the different subunits of DNA polymerase third with the help of this table. But you can see the structure is so complex and so effective that it increases the processivity of DNA to greater than 5 lakhs. So processivity of DNA greater than 5 lakhs is due to this beta sliding clamp which it moves effectively on it, and also the other two subunits core polymerases and clamp loading complex also assist in this process. So we can summarize the subunits which are present in DNA polymerase: alpha, epsilon, and theta. They are forming the core polymerase. Okay, so core polymerase is formed with the help of alpha, epsilon, and theta. So each is having two subunits. Okay, these are at their molecular weight, and these are different genes which are responsible for their synthesis. So alpha is having the polymerization activity, epsilon is having 3' to 5' proofreading exonuclease activity. Okay, theta subunits is not generally known, but it also does the polymerizing work, but with limited or low processivity. Then tau subunit which belongs to the clamp loading complex, it is having two subunits and synthesized by DNA X gene. Now, what they do? Stable template binding, core enzyme dimerization. Okay, so this is what stable template binding is. It is required for stable template binding. Okay, so that the strand would not shift here and there. So for stability, this unit is required. Now gamma subunit. This also belongs to what? That clamp loading complex. It is having one subunit. It's a clamp loader. Delta subunit, you can see it is a clamp opener. So clamp opener function is done by delta subunit, and delta dash subunit also having the clamp loader activity or function. So now the chi and psi, epsilon, which are not shown in the structure, chi interacts with the SSB. What this SSB stands for? Single stranded DNA binding protein. So it interacts with the this SSB protein. Okay. Uh, okay. So now then psilon, it interacts with the gamma and chi subunits. Now beta subunits which are forming the beta sliding clamp, they function for DNA clamp required for optimal processivity. So these three things are there, or three complexes are there in the structure of DNA polymerase third. The first one is core polymerase, second one is clamp loading complex, and the third one is beta sliding clamp. And each is having various different subunits for performing their function well. Now, enzymes involved in prokaryotic DNA replication. Now we can recall it, but there we have in the previous lecture we have given them name by simply helicases, topoisomerases, SSP, primase, and DNA ligands. But in E. coli and also in eukaryotes, they are named by different. Um, they are named differently. Like we have seen that DNA B, sorry, DNA B, DNA A, DNA G, they are what? Different names of these enzymes. Okay. So in E. coli, not only DNA polymerases, but also many other different enzymes are involved in DNA replication. So the whole complex of enzymes and proteins involved in replication forms the DNA replicase system of replisome. Replicase system, why we are seeing it as replicase system, because it function is of replication, and the whole complex of these enzymes and proteins will form a replisome. So the first important enzyme is what? DNA helicase. Okay, so this DNA helicase is there. It unwinds or separates the two strands of the DNA, and it also requires the ATP, energy. So in order to unwind the double-stranded DNA molecule, energy is required by which enzyme? DNA helicase. Now topoisomerase. So once the DNA unwinds or the two separate strands are separated, then it would also generate some torsional stress. Okay, so when you pull apart the two strands of a rope, what would happen at the two ends? Some strain would also generate, and that strain must be removed by some enzyme, and this enzyme is called as a topoisomerase. So it relieves the torsional strain. If that torsional stress is not relieved out, what would happen? The replication will not proceed smoothly, or it would happen. Now the third one is single-stranded DNA binding protein. Okay, once the strands are separated and both strands are what? They are complementary to their parental DNA strands, and they are bind with each other by hydrogen binding. So if in order to make sure that they would not re-anneal, there must be some proteins. They are called as a single-stranded DNA binding protein. Otherwise, what could happen if there is nothing in order to make them in separate position? Then what would happen? They will again re-anneal due to the formation of hydrogen bonds between them. So, in order to make them unwinded, to start the process of replication, single-stranded DNA binding protein, what it would do? It would bind to the two single strands or separated strands of DNA. It stabilizes the separated strands and preventing their rewinding. Now the fourth enzyme is primase. Why this enzyme is required? Is required for the synthesis of RNA primer. DNA ligase. The nicks are sealed by DNA ligase enzyme. So we know that on lagging strand, Okazaki fragments are synthesized, or DNA is synthesized in small pieces. So once the replication is completed, what would happen? The gaps would be there between those fragments. So that those gaps or nicks are sealed by the enzyme called as DNA ligase. So how it catalyzes or does the function of the ligating? It involves the formation of the phosphodiester bonds. And we know that phosphodiester bond would form at the 3' hydroxyl end of one DNA fragment and 5' phosphate end of the other DNA fragment. So once the bond between these two ends would form, that is known as a phosphodiester bond, and this bond is formed with the help of the DNA ligase enzyme. Now, the stages of DNA replication. So in E. coli, there are three different stages of DNA replication. The first one is initiation, the second one is elongation, and the third one is termination. Okay. Okay, now the initiation. Initiation means what? In order to start or begin, or we can say initiate the replication. Okay, so that initiation, we know that there must be a particular site or point at which the replication would start. So that particular point is called as the origin of replication, or we can also say it as ORIC. ORIC consists of, ORI stands for origin. Okay, the origin of chromosome replication. C stands for chromosome. Then consists of highly conserved DNA sequence elements, which is 245 base pairs in length. Okay, so this origin point is having highly conserved DNA sequence elements, and They are in 245 base pairs in length. So this stretched form is shown here, and this stretched form of of gene belongs to the origin of replication. So this origin of replication consists of some consensus sequences or conserved DNA sequences. And those conserved DNA sequences are arranged in two different repeats. So first tandem array is consisting of three 13 base pair sequences, while the second consists constituting of four 9 base pair sequences. These blue color is showing the four 9 base pair repeats in the origin of replication. And these four 9 base pair sequences provides the binding site for the DNA A protein. Okay. Now with the help of this diagram, we can understand the initiation of replication in E. coli at ORIC. So supercoiled template. Okay, this is what DNA. Sorry. Okay. This is what DNA of what E. coli. Okay. So it is having consensus sequences. This is what the origin of replication site. Okay. Now in this second figure, this origin of replication is stretched here. Okay. Showing the four known 9 base pair repeats and 3 13 base pair repeats. So this whole region belongs to the origin of replication or IC. So first priming or initiation is done with the help of 313 base pair repeats. Okay, I'm sorry. With the help of DNA A protein. DNA A protein are generally 4 to 5 in number. Here we are 5 in number, and it requires ATP also, energy also. So what it does, it binds to the three sorry this 4 9 base pair repeat sequence. You can see here this 4 9 base pair repeat sequence is in the form of a loop, and in that loop DNA proteins are there, so they are assisting it to form a loop. And in the next step, this is shown here that it would involve the um, for function of HU. HU stands for histone-like units plus ATP, also in order to unwind some portion of DNA. Okay, now in the next step, DNA B and DNA C. So DNA B, it acts for the helicase enzyme or is a helicase enzyme. So DNA C is helping DNA B to bind to these unbinded region of the DNA strands. So this DNA B helicase is a hexamer of two rings. You can see here one and two ring. So on the one parental strand, one DNA helicase is loaded, and on the second one, second DNA B helicase is loaded. Okay, so this is priming or initiation involves the separation or unwinding of the double-stranded DNA molecule. Okay, and it occurs in these steps. Now, we can summarize all the things which I have discussed in the figure here. So DNA A protein, about 4 to 5 molecules of DNA A protein binds to 4 9 base pair repeats in origin, and then it recognizes and denatures the 313 base pair repeats reaching 80 pairs. And this process requires ATP and bacterial histone-like proteins HU. Now second is DNA B protein or DNA helicase. DNA C protein assists DNA B protein to bind to unbound region. Here you can see DNA C is helping DNA B to bind to this unbound region. Okay, and this process also requires ATP. So two ring-shaped hexamers of DNA B proteins bind at each strands of DNA and then unwinds the DNA bidirectionally, and two replication forks are produced. Then single-stranded DNA binding protein, once the DNA strands are separated, in order to make them separate, means in order to make it not reanneal again, some proteins are required which are SSB proteins, which stabilizes the DNA strands. Then DNA G protein stands for the DNA primase, it synthesizes the RNA primer. DNA guidase, what it will do? It will relieve the torsional strain produced by DNA unwinding, and DAM methylase after the replication is complete. Both the parental strands would already methylated. So the newly strand synthesized, newly formed DNA strand must also be methylation of those newly synthesized DNA strand is also necessary, and it is done by the help of the enzyme DNA methylase, and it occurs at the 5' GATC sequence at ORI C. Okay, so once the replication is complete, DNA methylase would methylate the newly formed DNA strands, and also it methylates 5' GATC sequence at ORI C. So why it is methylating at ORI C? Here one thing is important. Okay, in order to start the replication cycle again, once the one replication cycle is finished, now the another replication cycle would start only when this enzyme methylates the 5' GATC sequence at ORI C. Okay. Now, these are the different proteins required to initiate DNA replication in E. coli. Okay, so already I have explained the function of these different proteins and enzymes. Once we can recall it, DNA A protein recognizes ORI sequence, opens duplex at specific sites. DNA B helicase unwinds. DNA C protein helps DNA B to bind to the unwound region of DNA. Then HU is a stone-like protein and it is DNA binding protein and stimulates initiation. Primase, which is DNA G protein, it synthesizes RNA primers. Single-stranded DNA binding protein binds single-stranded DNA. RNA polymerase facilitates DNA activity. Okay, DNA activity, DNA when recognizes the ORIC, then RNA polymerase facilitates its activity. Then DNA guidase, which is DNA topoisomerase 2, actually, it relieves the torsional strain generated by the DNA unwinding. DAM methylates once the one replication cycle is finished. In order to start the replication second cycle, the origin C should come in its previous situation of methylated state, so that methylation is done by the enzyme DNA methylase, and it methylates the 5' GATC sequence at ORI C. What is GATC? G-guanine, A-adenine, thymine, and cytosine. So these are the bases which are in that ORI C sequences. Now the elongation, which is the second step of DNA replication. This phase is marked with the synthesis of leading and lagging strand, and DNA polymerase third is the main enzyme that synthesizes the complementary strand. Leading strand, we know it starts with the synthesis of 40 to 60 nucleotides long RNA primer. So this is also important that the DNA synthesis would also require some primer in order to initiate the replication. Okay, so that RNA primer would provide the 3' end to the addition of the nucleotides by the DNA polymerase third enzyme. So RNA primer is synthesized by the primase or DNA G protein at replication origin. Now leading strand synthesis is in continuous in 5' to 3' direction along the movement of replication form. Lagging strand synthesis is discontinuous in short Okazaki fragments. So, but it also occurs in 5' to 3' direction and away from the movement of the replication fork. Okay, so we can say it this replication is going bidirectionally. Okay, and also it is semi-discontinuous in nature. Now, with the help of this figure, we can understand all those points clearly here. These blue color strands are of parental DNA strands. Okay. Okay, now on this strand you can see the red colored complementary strand, which is a leading strand. Okay, now on the other parental strand, lagging strand would be synthesized, but in short pieces, okay, of Okazaki fragment. So that Okazaki fragment would also require some primer in order to initiate the in order to initiate the addition of nucleotides to the lagging strand. So this RNA primer from previous Okazaki fragment. Okay, so this is a previous Okazaki fragment to which this RNA primer is attached. So in order to make them in unwinded state, SSB protein you can see here is present in the three um, tetramers subunits, and they are keeping those strands separated. Now here you can see this ring-like structure, which is what DNA B helicase and DNA primase. So these two primosome means forms a complex called as a primosome. Okay, these two activities are required here. So DNA B helicase, it will help in unwinding, while DNA primase will synthesize the RNA primer for the next Okazaki fragment. Okay, so here this green color structure is what once the replication would start, the leading strand is moving towards the replication fork while the lagging strand is away from the replication fork. It would produce some strain here, so that strain is relieved with the help of DNA polymerase second or DNA gyrase enzyme. Okay, so leading and lagging strand synthesis is shown here. Okay, see, you can see here that two Okazaki fragments are synthesized. Now for the next Okazaki fragment, RNA primase, DNA primase would synthesize a primer, and then to that next Okazaki fragment would be synthesized. Now here we can say that lagging strand synthesis is little complex compared to the leading strand synthesis. How it is complex? Lagging DNA G primase and DNA B helicase constitute a functional unit in the replication form called as primosome, where I am saying it functions as a replication unit here. You can see that DNA B and DNA primase both are together and they are forming a primosome over the lagging other parental DNA strand on which lagging strand would be synthesized. So primosome is required for the lagging strand synthesis on the other parental DNA strand. Okay. Now lagging strand synthesis occurs by loop formation. In this figure, you can see here, this loop is here and this loop is created on the lagging strand. So lagging strand synthesis would require the formation of loop. And why that loop formation is required? So that the replication on both the strands occur at the same rate. Okay. So for that, maintaining that rate same, that lagging strand synthesis would require the formation of a loop. Now, one of the two core polymerase subunits of DNA polymerase third is used in the synthesis of leading strand continuously, while the other subunit cycles from one Okazaki fragment to the next on the looped lagging strand. Now, understand it with the help of a diagram. See, this whole complex you are seeing here, it belongs to DNA polymerase third. So, this is what this is one core polymerase. This is second core polymerase. Huh? So one core polymerase is loaded on the leading strand synthesis, okay, and the other core polymerase is loaded on the lagging strand, or and the another parental DNA strand. Okay, so those core polymerases are helping in polymerization. Okay. Now the next point, DNA B helicase unwinds the DNA at the lagging strand as it travels in 5' to 3' direction. So this is the requirement for the DNA replication, that it would occur in 5' to 3' direction, and here DNA helicase, DNA B helicase would unwind the strand. Okay. Now what is there? DNA G primase associate with DNA B helicase at moments and forms a short RNA primer. Okay. Now you can see here. This is first. Okay. Uh, Lagging Okazaki fragment, which is synthesized on the other parental DNA strand. Now you can see the second Okazaki fragment is moving like this, okay, and it is traveling through the core polymerase. So this is synthesized, hence the core polymerase dissociated from it and is loaded on the for the synthesis of the another Okazaki fragment. Okay, once this is completed, what would happen? Primase. primase would come and join with the DNA helicase and will synthesize a new RNA primer for the synthesis of the another Okazaki fragment. Okay, so here we can see DNA primase binds to DNA B, synthesizes a new primer and then dissociates. Once the primer is synthesized, primase dissociates. Now, what would happen? These two segments are synthesized. Once this is completed, the third one would start the synthesis of next Okazaki fragment. Okay, till here the things are clear now. Okay, the other thing which is important, this red one is leading and lagging strand synthesis showing, and this black color arrow is showing the direction of movement of parental DNA through the complex. Okay, so parental DNA is moving in on the leading strand, you can see is moving towards the movement of the replication fork, while it is moving away from the movement of replication fork, and this arrow is showing the movement. Okay, so in this direction it is moving. Okay, so like this it will shift here. Okay, now at the primer, a new beta sliding clamp positions itself by the clamp loading complex of DNA polymerase third. Okay, at a primer, new beta clamp. Okay, so once this is completed, the first one is completed, it is completed. Now what would happen? It would dissociate itself in order to synthesize the next one. So you can see here, this is now free and now synthesizing initiated the synthesis of the new Okazaki fragment. Primer is here. Okay. So once this synthesis is finished, what would happen? That beta clamp which is present here is now dissociates from both the subunits, core polymerases as well as the clamp loading complex. From both the beta clamp is removed. Okay, and the new beta clamp in this figure, new beta clamp is added for initiating the another round of formation of DNA strand. Okay, so as the synthesis of an Okazaki fragment completed, replication halts and core polymerase or unit of DNA polymerase third dissociate the beta sliding complex and associate with the new clamp to start the synthesis of new Okazaki fragments. So once the Okazaki fragment synthesis is finished here, okay, the older clamps would dissociate and newer one would what added to the clamp loading complex as well as to the core polymerase in order to synthesize the new strand. Okay, so why there is a requirement for the new beta clamps? Because this polymerase is not fixed at one, it will move along the moving strand. Okay, it will move along with the synthesis of the leading and lagging strand. Okay, so it is positioning itself to the new place. From one Okazaki fragment to the new next Okazaki fragment, it is moving, or it is in order to complete the lagging strand synthesis, it has to move from one fragment to the next fragment. Okay. Now the RNA primer of each Okazaki fragment is removed after their synthesis. Okay, and is replaced by DNA with the help of enzyme called as DNA polymerase 1, and the nicks are sealed by DNA ligase. Termination. Termination means what? Now the replication is finished, now it has to be terminated. So termination also requires some sequence. So the two replication fork, okay, two replication fork which have originated from the replication bubble at the two sides, those replication fork must meet at some point called as a ter or terminus region, which contains multiple copies of 20 base pair sequences. The ter sequence function by binding to a TUS protein, which is called as a terminus utilization substance. The TUS-ter complex arrests the replication fork and replication stops. Okay, so for stopping the replication also some kind of sequence called as a ter sequence is required. The two replication forks which are moving in opposite directions and bidirectionally, they have to meet at some point, and where they meet is called as a ter point. Okay, and ter sequence require binding of some protein called as a TUS protein. This complex would arrest the replication fork and stops the replication. So in this way, the replication in prokaryotes is finished now. In the next lecture, okay, in the next lecture, I would uh, give my lecture on DNA replication in eukaryotes in full detail. So this lecture is beneficial for graduates, undergraduates, and as well as for postgraduates. Okay, so in order to get more video lectures in biology, please do like, share, and subscribe my channel. Okay, if any queries here, you can comment in the comment box, and see the playlist also on my channel for the previous lectures. Okay, thank you.